prokaryotes, just like all cells, divide by cell division. However, prokaryotes divide differently than eukaryotic cells. And the cell division in prokaryotes is called binary fission. And cell division in eukaryotes, which is a huge topic, which I'm going to cover later, cell division in eukaryotes is referred to as mitosis. So right now we're going to look only at binary fission. Binary means into two, and fission means splitting into two. And essentially, here are the steps. The steps are, first of all, you have to copy the DNA. So the loop that you see over here, this is supposed to be my DNA, and you can see that the DNA is connected over here to the side of the cell, to the membrane of the cell, and over here it is copied. So maybe I should actually indicate this by number 1, that this is now copied, and you see that there are now 2 loops. Now the bacterial DNA over here is pulled apart as the cell grows, and you probably can see already over here that this cell is a little bit larger than this one over here because it's growing. And as it's growing, the DNA, which is connected to the side over here, is pulled apart. And cells that grow have a lower surface area to volume ratio, and therefore they have to divide to restore the surface area again. And that's basically what it's doing in the next step. The cell starts to divide, and you can also see that it starts to be pulled in here, and a new cell membrane and cell wall start to form, and this process continues until you have 2 cells which are basically able to live on their own and to undergo the same process again. So we're back over here, and the DNA will again be copied. Okay, the second DNA, then it will be pulled apart, and then the cell starts to divide again. And that was basically it. There's not a lot more to say at this level. It's just that you have to know that prokaryotic cells divide by binary fission, and it's significantly simpler and less complex than cell division in eukaryotes, where you have many different many chromosomes that have to be correctly separated. But right now here we only have essentially one piece of DNA which is copied and then separated. So it can be much easier. As an interesting side information, bacterial cells, prokaryotes, bacteria are prokaryotes. Fast dividing prokaryotes can divide once every 20 minutes, every 20 to 30 minutes. Yeah, and I see that essentially that's number 1. Let's be correct about this. Number 2, it's pulled apart. Number 3, the cell starts to divide. And number 4, 2 cells are formed. And they might stay together or they might separate. It depends on the type of prokaryote. Okay. There is really not much more to say